Okay, I'm going to use this video to continue lecturing to the first cellular respiration PowerPoint. And I'm going to pick it up where we left off, which is at glycolysis. So I will cover glycolysis and the Krebs cycle today with this video lecture. So as an overview for glycolysis, you need to know where these processes take place. And this is the only process of cellular respiration that takes place in the cytoplasm. And in this process, there is a series of enzymes that will break down glucose, which is a 6-carbon molecule, into 2 smaller molecules of pyruvate, each of which is 3 carbons. And that does release some ATP. And it is also important to note that this process is anaerobic. It does not require oxygen. This is the only process of cellular respiration that can be considered anaerobic. Okay, we're going to start to go through detailed steps for glycolysis. This is an image taken from an older textbook that our class used to use. If you have access to any textbook or take a look at steps of glycolysis online, you would see something similar. So I will definitely emphasize what I feel is most important to take away from this. So we start with glucose. And you can see the structure of glucose here. Obviously, you should be able to draw the structure of glucose, but any of these other structures, I wouldn't really ask you to draw them out. It is nice to have them here in the PowerPoint because it gives you an idea of what's going on, but I don't expect you to memorize the structures of these. So really the first 3-4 steps of glycolysis, they're often called priming the glucose molecule to be split, or energizing the glucose molecule. And this, or it could also be called the investment stage. I've heard that as well. And what you're doing here, what's happening to the glucose molecule is this being phosphorylated by ATP twice and also being rearranged. So you take the glucose molecule, an ATP molecule is used to phosphorylate it. So we think of glycolysis as a way to make ATP, but in the beginning, ATP actually has to be used. So you're phosphorylating the glucose molecule at carbon 6. That's why it's called glucose 6-phosphate. There's the phosphate group added. Then it's rearranged to fructose 6-phosphate. You can see the carbon coming out of the ring. Another ATP is used to phosphorylate that, to put the phosphate group on another carbon. And now you have fructose 1,6-bisphosphate. So it has 2 phosphates. This contains more energy than the previous versions of the sugar. So at this stage, now this molecule is split. It is a somewhat symmetrical molecule, so it's split down the middle. And you get 2 molecules that look similar to each other. There's dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, G3P. But the DHAP quickly is turned into G3P. So really the end result of this steps 4 and 5 is to get 2 molecules of glyceraldehyde 3-phosphate, G3P. That is definitely a molecule you should know. You'll see it again and again in other PowerPoints. Now that you have 2 molecules of this, right? Because this was a 6-carbon molecule that was split into this 3-carbon molecules. There's 2 of these. Now you see with the image that there's a double arrow. So that means everything is happening twice for this molecule per glucose that you started with. So now you have an NAD plus being reduced to NADH. It's gaining electrons in a hydrogen atom and it becomes more energized. And that happens twice because you have 2 molecules of this G3P. Let's move to the next slide. Here's the next slide. You have 1,3-bisphosphoglycerate. And now the phosphates that are on the molecule are going to be added to a molecule of ADP to make ATP. So you have 1,3-bisphosphoglycerate. It phosphorylates the ADP to ATP. And again, this is happening twice because it's per glucose molecule. And you get 3-phosphoglycerate. And then that's rearranged to 2-phosphoglycerate. And then finally, the final phosphate is added to an ADP molecule to make ATP. And then you end up with 2 molecules of pyruvate. That's the end product of glycolysis, along with the ATP and the NADH that is made. So key parts of the process for glycolysis: 2 ATPs used to prime the glucose for splitting. The fructose 1,6-bisphosphate splits into DHAP and G3P, but that DHAP is converted to G3P. 2 NADH are formed just after that step. 2 ATP are formed by substrate level phosphorylation in both steps 7 and 10. And 2 pyruvates are produced at the end in step 10. So if you look at a yield of products, we have 4 ATP produced, but 2 are used in the beginning. So glycolysis has a net production of 2 ATP. You also have 2 NADH and 2 pyruvates produced. And these molecules are what move on to further processing if oxygen is available. And that's why it's called aerobic cellular respiration. So that's glycolysis. A little bit more about the energy that's created. These 2 molecules of ATP, if you want to quantify how much energy they possess, is 31 kilojoules per mole. That gives you 62 kilojoules of energy from a possible 2870 kilojoules that are in a glucose molecule. So you're not really getting much energy out of the glucose molecule by doing glycolysis, only about 2.2%. Most of the energy is still trapped in the bonds that make up the pyruvate molecule and the added electrons that were passed to the NAD plus to make NADH. But there's also some that has been lost as heat. It is thought that the earliest cells on Earth, because they were all prokaryotic, they did not have mitochondria, and there was not a lot of oxygen around, these cells used glycolysis to get their energy. And glycolysis was able to provide enough energy to these cells because they were only unicellular. There are still plenty of organisms, simple organisms, that use glycolysis for energy today. But any multicellular organism uses the rest of cellular respiration, the Krebs cycle electron transport chain, to produce most of the ATP. But they still do glycolysis, but it takes place in the cytoplasm. So I'll quickly move on to pyruvate oxidation. And notice that this happens if oxygen is present. So the following is going to occur to the pyruvate molecules, because there's 2 of them that enter into the pyruvate oxidation from glycolysis, because everything is per glucose molecule. So you're going to have a CO2 removed from each pyruvate. So it goes from a 3-carbon to a 2-carbon. The NAD plus is reduced to NADH, and that remaining 2-carbon compound becomes acetic acid. Then a coenzyme A is attached to the acetic acid to form acetylcoenzyme A. So this is not just, even though it kind of alludes to it in the PowerPoint that it's one step, it's actually multiple steps that take place on one enzyme complex called the pyruvate dehydrogenase complex, which is really 3 enzymes. So it's a little bit more complicated than what we have here, but this is what I want you to remember. Here's an image showing it. Please make note that the pyruvate was produced in the cytoplasm, and it's got to get into the matrix where these reactions occur. So the pyruvate has to go through a transport protein. There's the CO2 that's removed to make it a 2-carbon molecule. There's the NAD plus being reduced to NADH. There's the coenzyme A being added. And you get the acetylcoenzyme A, which is the final product of pyruvate oxidation. Here's the energy yield, 2 NADH, 2 acetylcoenzyme A, 2 CO2 molecules released as waste. Just a little bit more about acetylcoenzyme A. It comes from vitamin B5. Carbohydrates, as you saw in the previous slides, can be broken down to get acetylcoenzyme A, but so can proteins and lipids. And we will look at that in a further PowerPoint. The acetylcoenzyme A can be used to make fat or ATP. And really, the concentration of ATP in the cell will determine which pathway the molecule will take. If the cell is not using that much energy and it has enough ATP, then the acetylcoenzyme A would be used to make fat. Additionally, what happens to acetylcoenzyme A will depend on whether or not there's oxygen present. If there is, it's going to move into the Krebs cycle to undergo further aerobic respiration. If there's not oxygen present, it will become lactate or lactic acid, which is anaerobic respiration. Here's what the molecule of acetylcoenzyme A looks like. Not that I would expect you to memorize that structure. And that is the end of the part 1 PowerPoint for cellular respiration. We will cover Krebs cycle and electron transport chain in the next video lecture.